This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode 37 for June 19th, 2009. Hello, everyone. I'm Vincent Racaniello. It's time for another exciting episode of TWiV. And joining me today, Alan Dove. Hello, Alan.
Hello. Good to be here.
Alan's coming in via Skype. And a newcomer to TWiV, Eric Freed. Welcome to TWiV, Eric.
Thanks. How are you doing?
This is our first guest from the National Cancer Institute. Eric is head of the Virus Cell Interaction Section, National Cancer Institute, which is in Frederick, Maryland. Is that correct?
That's correct, yes.
Eric works on HIV, and he's also editor-in-chief of Viruses. And Viruses is a new open access journal, so I thought it was time for us to talk about open access on TWiV. So we'll get Eric's thoughts on that later on, and perhaps if we have some time, we can also talk about your lab work. And,
okay.
And if we don't get to it, we'll just have you back another time.
Sounds good.
First story is Alan's Genzyme is shutting down manufacturing of 2 enzyme drugs because of Vesey Virus contamination of their bioreactors.
That's right. Yeah, and I initially saw this, one of the papers I read, or at least it will be a paper until they finish going out of business, is the Boston Globe. And
going out of business, is that right?
Oh, yeah. Yeah, they're owned by the New York Times, and the New York Times is trying to shed some debt, and that's one of the things they're thinking of getting rid of. But that's not very virological. What is the they cover obviously a lot about Massachusetts businesses, and Genzyme is a big one. So Genzyme's a big biotech company. They make enzyme therapeutics, and they have had to shut down production of two of their drugs, Cerezyme and Fabrazyme, because they had contamination of their bioreactors with Vesey Virus. And this was big news to me because I had never heard of Vesey viruses, and so I immediately tried to find out more. And hopefully, Vince, you'll be able to fill us in on exactly what these things are. But the upshot is that they're gonna have to shut down production of these 2 enzymes for several months, and this is a big deal because, as you may know, in enzyme manufacturing, usually getting enough of the product is a big problem. So they're usually maxed out on production capacity, and patients who are already taking these drugs are hopefully going to be able to have enough of a supply, but it's a major issue from the corporate level all the way down to the patient level.
This is very interesting. So this is a contaminant of CHO cells that they use to produce the drugs, the enzymes that they make. CHO meaning Chinese hamster ovary cells.
Right.
And so I did a little searching. So Vesey virus is a member of the Caliciviridae, the same family that has the noroviruses in it.
Right.
So these are RNA viruses, positive stranded with an icosahedral capsid, no envelope. The Norwalk-like viruses are the ones often implicated in gastroenteritis in humans. But this family has 4 genera, and one of the genera is Vesey virus, and that is where this virus falls in. It's in this genus, and Vesey is named after vesicular exanthem of swine virus. So the vesicular part, that was one of the original viruses that was discovered in this family. And it's a very interesting family. It contains a number of interesting viruses, cow viruses, whale caliciviruses, primate reptiles, and San Miguel sea lion virus. So you have terrestrial viruses here, and we also have oceanic virus. So this Vesey virus, what's the number? 2112?
2117, I think.
So this was discovered in 2003. There was a paper in the Journal of General Microbiology, identification of a Calicivirus isolate of unknown origin. So this lab in Tübingen was growing CHO cells, and they started dying. So they looked at them by EM, and they found virus particles in them. And they isolated the virus and cloned it and sequenced it. And it was this, they called it Calicivirus 2117. And the sequence shows that it's a member of the Calicivirus family. And that's pretty much all there is on this virus. So it's a Vesey virus by homology. They put it in this family, with the other San Miguel and the whale viruses, but not a lot known about it at all.
Are these widespread contaminants of CHO cells? Do you know? That's a very commonly used tissue culture cell line.
Yeah, I don't know. They say they have no idea where it came from. It just appeared. And I think in some of the press releases that I read, they mentioned that this is a common contaminant of CHOs.
Well, it's certainly common for them.
Yeah, it's unfortunately common, but I found nothing that would give any indication of, how widespread it is.
Right.
The interesting thing is, where did it come from? I mean, this is a cytopathic virus, so it's very obvious when you have it.
Right.
And, in the lab in Germany where it was originally isolated, I just don't know. I mean, unless there are conditions where the virus is, the genomic material is there, but it doesn't replicate, and then at some point. But I would think that they test these cells for a variety of viruses before they use them.
Yeah, when they're gearing up for enzyme production, I know the hoops that you have to jump through to do that are pretty impressive. So they would have tested these cells for pretty much everything, and also obviously for viability in growing them up. And I would think that any contaminant in that process would have shown up if it was something that would manifest itself.
Here I have it. This paper says fetal bovine serum is the most likely source of contamination.
Oh.
So they have, this is related to cow caliciviruses. So they developed a PCR assay, are screening it, but they don't say whether they found it in any batch of fetal bovine serum.
Any indication as to whether this virus from that source is actually infectious for humans?
Yeah, there's a statement in the Genzyme press release that says this Vesey Virus 2117 has not been shown to cause human infection, which I think is an interesting way to phrase it.
Yeah, it doesn't mean that it can't. It just means
right. It doesn't mean that it can't. It just means we don't know that it does.
Yeah.
So that is certainly a concern in this. Is if you're growing up these enzymes, you don't know how long the virus has been there, and maybe some batches went out with this. Now, whether that's the case, I don't know. I'm sure they're now in the process of running that down and testing the batches that they made before this and seeing when the contaminant showed up.
I think fetal bovine serum makes the most sense, right? Because that would explain it suddenly popping up in your cells.
And that's probably the least controlled component of your entire process, too.
Yeah, which is a good reason to try and get away from that for all kinds of production, right?
Yes.
I just read an ad in Microbe, the ASM publication. It said, if you can't wait to get away from meat in your cell culture, here's the solution. So, I mean, it's kind of weird that after all these years, we're still using a pretty crude animal product, right, to grow human enzymes, virus vaccines even. I guess it's just very expensive to do the serum free for production, right, to have hundreds and hundreds of liters, so...
Oh, yeah, the scale of these things is just enormous.
But I guess what you could do is take every batch of fetal bovine serum and screen it for every known virus.
Yeah.
Be expensive.
Except, that would be expensive. I'm not sure if that would compare cost-wise with going serum-free, which would get you past the unknown viruses, too.
Yeah.
Related question is whether heat inactivation, which is often used to prepare fetal bovine serum for tissue culture work, actually inactivates this virus. This is a non-envelope virus, so I imagine it's relatively insensitive to the typical 50 to 60 degree Celsius heat inactivations that are usually used.
Yeah, it's a good question. Why are those done anyway? Do you know, Eric?
I think to inactivate complement.
Complement, right,
yes.
Yeah. Actually, we stopped doing it because we don't work with enveloped viruses. I figured we don't need to inactivate complement, so... But maybe it's a good idea to resume just to get away from this possibility. So it's possibly from the serum. That's very interesting. So they've shut down. They have to clean up and get started again.
Yep.
So that's an interesting example of how you can get contaminants if you still use these kinds of products, if in fact that's where it came from.
Yeah.
Quite an interesting family of viruses.
It's interesting to think about the transmission of viruses that live in these very different environments. Think about viruses that have to transmit in saltwater versus viruses that have to transmit through air, et cetera. It's very interesting mechanisms of spread.
Oh, the ocean viruses are fascinating. I don't know if
Yeah.
you've ever read much about them, but I'm amazed
Yes,
at
very interesting.
this question of how they transmit. I mean, how do whales transmit viruses to each other? I guess they excrete so many. I think it's 10 to the 13 particles per gram of feces, and I think whales put out a lot of excrement.
I imagine they do.
So there's a lot of viruses. And I mean, whales are not typically group animals, right? They don't hang out.
Oh, they do.
They do?
They many species of them travel in pods. And
What did you say? They have
pods.
They have iPods?
Yes. Well, they've got to listen to something besides all those whale songs.
So that's how it could be transmitted among the pod. Okay.
Yeah.
Good thing this is not this week in
Cetology.
Cetology, yeah. All right. Well, that's cool. I like that story, Alan. Our next story, I don't know if you saw this, guys, in the past week. Well, of course, every day there's something about H1N1 in the news. It's like every death has to be documented and every new isolate has to be written about. So there was an isolate from Brazil, and a couple of papers claimed that this had new mutations that would allow the virus to spread to new hosts. And so it turned out that the sequence was in the data in NCBI, as most of them are. So I had a look at it. And in fact, it's identical to about half of all the isolates that are out there already. And the other changes compared to the other isolates were meaningless. So I think I give the journalists the benefit of the doubt. I figure they misunderstood the Brazilian scientist. And he said, we're looking for changes that could affect host range. And they said, he's got changes that affect host range. Isn't that the way it works, Alan?
Pretty much. If you've got somebody who's on a tight deadline and they're getting a story out and what's this about this Brazilian case, and you call up a scientist and there's some blame to share here, I'm sure.
Shouldn't they check their facts, though? I mean, typically you do that, right?
You do to the extent that you have time and ability. But if you're talking to a scientist who seems to be an expert on this, and he says, well, yes, it has changes from the other virus, I would call it, you could certainly define it as a new strain because it's different. And then you go to press with that, and there you go.
A couple of weeks ago, there was a New York Times article about changes of this H1N1 virus, and they quoted Ian Lipkin as saying that virus doesn't want to get too lethal, And so I wrote a blog post saying, why not? How do you know? And he emailed me, said, they misquoted me. I didn't say that.
See, that's the other thing, is the researchers will always say they were misquoted. We don't have that luxury here on the podcast, though.
No, well, we get corrected, though, and that's fine. I don't mind being corrected. It's okay. The interesting point about all this is that everyone is looking for changes that might lead to increased lethality, right? Because, back in 1918, there were these waves of the pandemic with increasing virulence. So everyone is just thinking, well, is this going to happen with this virus? But, if you go back and look, there's no evidence from any pandemic that the increased virulence has anything to do with mutations in the virus. There's no data. In 1918, we only have one isolate. And I asked Peter Polese about the 57 and 68 pandemics. He said, nobody's looked. Take samples from different parts of the pandemic and look at them in animals and look at the sequence. Nobody's done that. But here, of course...
Somebody should do that.
Yeah, here now with this one, we've got an isolate a day, right?
Sure.
This is going to be the most documented influenza pandemic ever, I'm sure.
Yeah, in all ways. I mean, the bioinformaticians alone are having a field day. In fact, I'm amazed at how many papers are published just on analysis of the sequences. It's incredible. I guess where you are at NCI, there's no influenza lab, right, Eric?
Not in the NCI. There's a little bit of interest in seeing what can be done about influenza here in the NCI, but it's not obviously a major focus.
Right. It's not. It's over at NIH campus, I guess it would be.
Yes, that's right.
I also found a nice map of influenza on the WHO site. They made a new interactive map, and we'll put a link to that in the show notes where you can see total cases and where they are in each country. And I just wanted to note that since the last TWiV a week ago, there have been 10,000 new confirmed cases of influenza globally. So it's certainly increasing substantially.
Those are H1N1s or general?
Those are H1N1s, yeah. Australia, South America, I saw also South Africa is now confirming cases. So it's moving on in the Southern Hemisphere. But we still seem to have some here in North America. The U.S. still has cases. The CDC site has a graph of their isolations, and the H1N1 is still rising. So I'd be interested.
Even though it's off-season.
Yeah, it's quite interesting. So I guess the pandemic strains often break the rules about seasonality.
Well, and as we commented last week, there's also the issue of increased monitoring.
Yeah.
So we're probably seeing cases that we never would have seen before.
Yeah, in fact, there was an article this week somewhere where they said one of the benefits is that you get to see what strains are circulating during the summer.
Right.
So usually CDC stops surveillance in May, right? So for now, we're going to go probably through the summer, and we can see how long the strains circulate. I think it's very interesting. I think from what I saw on the CDC site, the old H3N2 and old H1N1 strains are now very, very minimal fraction of the total isolates. Seems to mostly be the novel H1N1. There are all kinds of hypotheses, but you can imagine that when you have a lot of population immunity, maybe that helps the seasonality. It helps get rid of the virus over the summer. But this new strain, since very few people are immune, doesn't behave in the same way. Before we go on to our other two stories, here's a word from our friends at Citrix, the makers of GoToMyPC.com. As you know, and I've told you many times before, I rely on my office computer. I would be lost without it or GoToMyPC. With that software, I can access my computer from any internet-connected machine. So that means I'm not stuck at the office. I can be productive from wherever I am. And GoToMyPC is the only remote access service I recommend. You'll love accessing your office PC from home, and one of the best things is how easy it is. You log on to GoToMyPC.com, and your PC desktop instantly appears. I can use any program, work on any file, check email, access network resources. It's really been a life-changing solution for me, and all I want now is to be able to use it in my car.
How about the beach? It works there too, I bet.
It works at the beach, that's right. Yeah, wherever you are. But the car is where I spend most of my life. And if I could, I know I could probably get internet access by the card that you were trying to use before, Eric. But
yeah, certainly.
I have to drive, so I can't be doing that.
Well, if you could take the train for part of your commute, then you could do it from there.
Yeah, the train would be good. Anyway, you can try GoToMyPC free for 30 days, but you must visit GoToMyPC.com/podcast. That's GoToMyPC.com/podcast. You'll get a free 30-day trial. Tell them TWiV sent you. I saw on Twitter the other day, someone heard them playing TWiV in a Starbucks over the speakers.
Boy, I bet that drove business out, huh?
The guy who did the tweet, he said everybody was having a really animated conversation about viruses. Can you imagine? Alan, we have made it. We're in Starbucks.
That's amazing. We're in Starbucks. Well, hey, if that's true and you're listening to it right now, you should go get yourself a double extra grande latte, whatever it is, because that's certainly something you should support.
Usually when they play the music in Starbucks, you can buy the CD right next to the cashier or download it from iTunes. But maybe we should have a TWiV CD there so they could buy it if they want. I thought that was great. It's TWiV in Starbucks. Alan, computer models show that targeting children maximizes the effect of vaccination for diseases such as influenza.
Yes. Now, targeting children, you have to be careful when you talk about that. But
we're not hurting them, right?
No, we're not hurting them. We're targeting them with the vaccination. So these are a couple of researchers at University of Warwick in the UK did this computer modeling study. This actually came out in May, but I just came across my radar recently. They built, I gather this is primarily a demographic computer model where it wasn't so much the accounting for the increased rate of infection among kids, although they may have done that, but they were just looking at how many households have kids in them and model from that and how many other kids are they contacting. And of course, children go out and they have contact with all their classmates at school every day, which is an unusually good environment for transmitting infection. So in this model, they tried various scenarios, and they determined that if you had a limited supply of vaccine and you wanted to look at distributing it most effectively, they found vaccinating the children was highly effective. And it generally outperformed random vaccination. And it also reduced the amount of variability you had in epidemic transmission between different regions of the UK that they were looking at. And this is not, the result is not terribly surprising. Certainly, if you have kids, you understand that they are extremely good at transmitting infection, and they're probably the ones you'd want to target in controlling something like influenza. But it's good to see a modeling study like this backing that up and really, delving into the types of effects that you might be able to accomplish by specifically setting up flu vaccination in schools, for example, and looking at that population in particular.
So this is a modeling study. They didn't actually look at specific infectious diseases, right?
No, right. That would be the next step, of course, would be to go and look at. Yeah, there are certainly infectious diseases where you could probably do this, although, if you look at something that's vaccinated in childhood, then most people would have lifelong immunity, right?
Yeah, sure. It's interesting. They look at the demographics, the household structure, how many kids, and what age they are and where they go and so forth. It's very interesting, and it makes perfect sense, as you said. And of course, we knew this, having had kids, that you should immunize them for sure. I think it's interesting, particularly in terms of the anti-vaccine movement, which of course is targeting children. So this is another opposing view, right?
Which would, and the anti-vaccine movement in targeting children is going to maximize the opposite effect. So that's, by doing that, they are maximizing the number of casualties that they're going to cause.
Yeah. Eric, if an HIV vaccine were ever made, would it be something given in childhood, do you think, or would it be just for at-risk populations?
Well, I think it would depend a lot on the characteristics of the vaccine, the longevity of the imagen, and so on. I think ideally a highly effective vaccine would be given in childhood, and with whatever boosting would be necessary during the adolescent period. But I think we'd really need to look at the actual type of vaccine that were produced, if that in fact ever is possible.
Yeah, that's the other question, of course, right? But I would imagine that would make the most sense. But, in the old days, we made vaccines that didn't last very long, and the first polio vaccine was quite short-lived, and it was given to all kids anyway.
Right, because that's really a childhood disease to a large extent, and HIV is really a disease of either children getting it during, in utero, or of course when they reach sexual maturity.
So between birth and sexual maturity, there's no significant incidence.
There's transmission through milk
right
during nursing, but beyond that, no.
Yeah, so presumably at some point you have all the mothers immunized, and that would take care of the in utero spread.
Right.
Mm-hmm. Yeah, so it might make more sense to deploy it kind of like the HPV vaccine.
That's right. I think that's probably what would, and of course initially they might target at-risk populations rather than globally, but of course that remains to be seen, and we're still lacking a vaccine candidate, so that's just speculation at this point.
Sure.
Right.
Yep, we are. How far off do you think we are, if at all? You have
well,
any sense?
The horizon seems to continue to be moving further out. I think at the moment there's probably a greater degree of pessimism than I think I've witnessed really in the last decade. I think people are really not very positive, and of course there have been major strides made in treating HIV-infected patients with drugs, with another very effective drug, the integrase inhibitor, coming online very recently. So I think one route has really been very successful, and the other approach really so far has failed. But obviously, globally the vaccine would really be the way to go ultimately. So
that's actually one of the most common things that comes up in conversations with lay people, as I'm sure you've encountered. You always get the question, well, you worked on viruses. Why don't we have a vaccine for HIV when we've got vaccines for all these other viruses? And it's obviously been an extremely difficult road.
That's right. Yeah.
Well, since we can't understand it for the most part, it's hard for everyone else to, right?
Right, exactly. It's a very hard question to answer.
Our last story has to do with a virus called chikungunya virus in mosquitoes. And I don't think we've ever talked about chikungunya on TWiV, have we, Alan?
Despite the wonderful name.
So when I was a graduate student in the 70s, I thought this was just the greatest name, chikungunya.
It's great, yeah.
And I remember reading about it, and it was quite it was an exotic virus, let's say. A virus identified in 1953, an enveloped mosquito-borne RNA virus, primarily a third world disease, which was pretty restricted, and apparently the restriction was because of the mosquito vector, Aedes aegypti.
Right.
But in 2004, it started moving. It moved to Europe and could get into the U.S. So I think there was an outbreak in Kenya. It spread to Indian Ocean islands, Réunion, one of those. And on Réunion, there was a big outbreak. 40% of the population, which is about 800,000 people, got infected. Went to Italy and India. And, of course, people coming back to the U.S. from those countries have even brought it in.
And this is a virus you don't want to get, as I recall.
It's not very good. Severe rash, joint pains, not always fatal, but not something you'd like to get, right?
Right.
The interesting story here was that the expanded range of the virus apparently came about because it got into a new vector, Aedes albopictus, the Asian tiger mosquito. It turned out, it's a very nice paper a number of years ago now, showed that a 1 amino acid change in the viral glycoprotein accounts for the more efficient replication in albopictus.
Evolution at work.
Amazing. So the mosquito previously was never a good host, and now this 1 amino acid change allows the virus to grow much better in this mosquito. And as we talked about just last week, I think, albopictus is now throughout the U.S. It was brought in spare tires on cargo boats.
Right.
And so there's the potential that this virus infection, chikungunya, could be in the U.S. as well at some point.
Is there any information about what this single amino acid change does to mosquito to human transmission and pathogenesis?
I don't believe so. I think the main information is that it's now in a mosquito that has a broader range.
I see.
And in fact, the paper that the paper I'm talking about today is a PLOS paper. It compares the replication of the virus in the two different kinds of mosquitoes, albopictus and aegypti. And it basically finds that albopictus is slightly better replication, but not significant. So really, it's the expanded host range of the tiger mosquito that's responsible for the spread.
I see. Mm-hmm.
And the strain that's adapted to albopictus seems to, I don't know if it may have some slight problems, but it certainly does seem to transmit pretty well.
It does transmit pretty well, yeah. Now, this paper I thought would be interesting because it's experiments in mosquitoes. So they basically infect mosquitoes and assay viral replication in the mosquito, and they also measure the virus in mosquito saliva. Now, these are not techniques that I or you, any of you probably have ever done in virology. We grow viruses in cell culture and titrate them in cells. But I thought it would be interesting to just briefly go over what they do. So they have mosquitoes that in the laboratory, which they purchase, and actually they got these mosquitoes from La Réunion, which is the island where the outbreak was in 2004. They feed them virus, and they put the virus in a buffer, and they add red blood cells to it to get the mosquitoes to drink it.
Right.
So they put this mixture of red blood cells and virus in the cage so that the mosquitoes can feed on it, and they give them 15 minutes. After 15 minutes of feeding, engorged females were sorted on ice and transferred to cardboard containers. And then they assay virus replication. They do PCR at different times after infection, and they compared the two different mosquito strains. And then they also measure virus in the saliva. And this I thought was amazing. So they take the female mosquitoes, take off their wings and legs, and then they put the proboscis into a 1 microliter micropipette. okay, which has some bovine serum in it. And then they apply acetylcholine analog to the thorax to stimulate salivation. So they collect the saliva for 45 minutes, and then they go and they assay for a virus. This is just amazing because nothing I have ever done, obviously. But can you imagine you're putting a tiny pipette, the proboscis, into that and wait? I guess that's why they have to take off the wings and legs so they don't move around and disrupt this process.
Yes, so they don't leave.
But these mosquitoes are alive, and they stimulate them to give saliva. And then they check this out, and they do the titer of virus in saliva, PFU per ml. Amazing.
That's incredible. They must need very good eyesight, people to do that work.
And very patient.
Yeah.
So it's for us, it's easy to infect cells, but in a host like this, I infect mice, of course, and I think that's quite easy. But a mosquito, of course, I couldn't do this anymore because I can't see well enough to do things in mosquitoes. So you have to have young people doing this. But it's very nice work. We'll put a link to this in the show notes so that you can see just how this is done. And of course, the conclusion is that the Albopictus is slightly more efficient than a gypti to transmit the variant because it has a little bit more replication and has more in the saliva. And the other interesting thing here is that 2 days after infection, they can find the virus in the saliva. So it happens very quickly.
Which is probably about when the mosquito would be feeding again.
Oh, yeah?
Yeah.
That's right. So there you go. It's adapted to be effectively transmitted, right?
Yeah.
Okay, so that is chikungunya, the virus with the great name. And that brings us to one of the things I wanted to talk about with you, Eric. You've become an editor-in-chief of a new journal called Viruses, right?
Yes, that's correct.
And this is an open access journal. So maybe the first thing we should ask is, why did you decide to take this on? You have an active lab, right? So why did you decide to take this on?
Well, it's actually a good question, and it's something I had to sort of mull over a little bit. So initially I was asked to be on the editorial board, and that wasn't a big deal, so I quickly agreed to do that. And then the journal later approached me to be editor-in-chief, and I had to really give that some thought. As I'm sure most listeners know, there are quite a few virology journals, many of which are very well established. So it's really hard for sort of a startup journal to really make a mark in this area. But then again, thinking about it a little further, I realized how important viruses are. Really, many of the things we've talked about today, and I'm sure in all your other shows. So it seemed like a good opportunity, and the open access for listeners who aren't familiar with that, we alluded briefly a moment ago to the Public Library Science Plus family of journals. The concept of open access is basically that anybody, anywhere in the world who has internet access can log on and access these journals, access all of the articles in those journals as immediately after they're published, completely free of charge. So there's no expensive subscription, or you don't have to be a member of a professional society, or you don't have to work at a university or at a research foundation. So it's really science that's accessible to everybody, which I think is something that probably the listeners of this show would really appreciate.
Yeah, there are many cases where we have papers that we'd like to share, but not all the journals are open access, and people realize that. So if I'm not mistaken, the idea of open access also means that you can take the work, the paper, and modify it as long as you give credit, right? So you could take figures and use them. It's sort of like open access software, if I'm not mistaken. Is that correct?
Yeah, that's correct. The only exception to that is if a research article in an open access journal reproduces a figure from another journal, then the copyright of that figure actually originates with the original publication,
right?
Not the open access journal. But apart from that, yes, everything that's published in terms of figures and information, provided proper credit is given, can be used in teaching, can be further reproduced in other publications.
Yeah, I often use figures from these journals on my blog because I know I don't get in trouble when I do that. So what was the first open access anything? Was it software? Do you know?
I don't actually know. I would imagine that software probably goes way back, maybe some music, although of course that's been very controversial.
Yeah.
But this is something that, among others, Harold Varmus really pushed when he was the director of the NIH, that basically the taxpayer really foots the bill for the research that's done in this country to a large extent, and therefore should have free and easy access to the results that come from that research. So I think it's a good idea. There are concerns, though, associated with open access, and particularly the proliferation of open access journals. And some of you may be familiar with a recent story in which a graduate student at Cornell basically essentially made up a paper. The paper was completely nonsensical, and he sent it to one of these new open access journals, and they claimed that it was peer-reviewed and it was accepted for publication. This is an article that, if anyone had actually read it, they would have quickly realized that it was complete nonsense, and really the authors of this paper did this really to make a point that peer review is really a linchpin of scientific publications, whether it's open access or otherwise. And one thing I've done as editor-in-chief, or try to do, is to really be absolutely certain that in this sort of accelerated publication process that accompanies open access, we haven't sacrificed any of the peer review process and the rigor of the data that we publish.
I think some of the concern of that initially arose because of the economics of open access. I mean, listeners may not know how you keep the lights on when you're giving away all the research for free. But in fact, the people who are publishing the paper are paying a page fee, right?
Yes, and that is, in fact, I think what was happening here is the organization that was publishing this was basically charging, I think, something like $800 per paper, and basically subjecting it to what in effect appeared to be no peer review whatsoever, and then, of course, collecting the fee. Our journal is actually free of charge for 2009. That is, you can publish papers this year without a charge, and then beginning next year there'll be a fairly nominal charge that will just help defray the cost of running the journal.
So this false paper was done as a way of testing the system, I presume.
That's correct. Yes.
All right, what are there other open access besides PLOS and viruses in our field? Are there other open access journals?
Well, the organization to which this fake journal was submitted is Bentham, and they actually publish more than 200 open access journals. Many of these quite new, and there's at least one of them that I actually happen to be on the editorial board for, called the Open, TWiV. So that is a virology journal.
Mm-hmm.
But so there are other virology-related journals that are open access. But as well, there are many very well-established journals like Journal of Virology and so on that are the more conventional non-open access. Although I think journals in general are becoming more and more open access. For example, those of us at the NIH have to post our accepted publications online for everyone to access. So I think there's definitely a trend in the scientific publication field to make the results of the research available free of charge to everyone.
Right.
I was at a forum, I think, a couple of years ago where we were discussing this. It was a virology meeting, and the idea that the government foots the bill for most of research should make it available to the public. But then there were some people said, well, universities also pay money into this, and it's not all government supplied. So what do you think about that?
Yeah, I mean, that's certainly true. And of course, there's a lot of private sector contribution to research. So this is something that is continually debated, and certainly the publication establishment is somewhat opposed to the concept of open access. I mean, it does cost money to publish things, but but in any case, I think journals like Viruses gives people that option if they choose to pursue it, and it does really streamline the publication process, makes it much quicker. In fact, we just had a paper that was published 7 days after it was submitted, and that's a record for the organization that sponsors Viruses, which is an organization called Molecular Diversity Preservation International, or MDPI. And we did submit this paper to rigorous peer review. We told the reviewers that this is something very important and timely. It actually deals with H1N1 outbreak in Milwaukee, Wisconsin. So it did get rigorous peer review, but we did manage to get it out within 7 days. So I think that's another benefit of open access, or at least online journals. The process can actually be very quick, which sometimes is important in fast-moving fields.
But that's not unique to open access journals, though.
No, that's true. Right.
Yeah, in fact, I got that paper by email just the other day, the H1N1 in Wisconsin. And the nice thing is I can share it with our listeners. There's no problem.
Yeah.
Don't have to worry about it. So aside from people maybe submitting things that aren't correct, which can happen anywhere, are there any downsides to having open access publications?
If you're looking for the beautiful, glossy, hard copy of an article, that does cost money, and you won't find that so much with the open access journals. But I think in general, I don't see any major downsides. Maybe
in terms of the finances. I mean, I don't mean to harp on money, but this does seem to be an issue that open access has struggled with. It does cost money to publish a journal, and
right.
Obviously you get a certain amount of free help from the peer review process, which is something that a lot of the closed access journals have been rightfully criticized for, claiming that they're providing this service when in fact the peer reviewers are working for nothing.
As is the editor-in-chief in this case.
As is most of the editorial staff.
Yeah.
But somebody has to lay out the pages. Somebody has to maintain the server. There are things that do need to be done, and I think one of the concerns that's come up frequently is, well, how do we know that open access journals are going to be sustainable over the long term? At 10, 15 years from now, will PLOS still be in business? Or given that, I don't know what the financing is like at Viruses, but I know PLOS still relies substantially on donors to finance the operation.
Yeah. No, I mean, I think it's very much like the proliferation of small biotech startups. There are many of them, and a large number of them don't survive, and I think only time will tell how successful any particular journal is, and in fact how successful the effort is. I'm not sure that the virology field can really support a large additional number of journals, but I think given the activity in the field and the number of publications being submitted for publication, I think there certainly is a demand for certainly a reasonably high volume of publication. But you're absolutely right. Not all will survive, and I think only time will tell.
Now that brings up another issue that I occasionally, I just in case listeners are getting the wrong idea, I'm actually really strongly in favor of open access journals. I love being able to just go and download the entire paper, and it frustrates the heck out of me when I can't, when I'm researching a story, for example. But I do worry that this is going to end up being like during the .com boom, when you could have Cosmo.com delivering a candy bar to your door, if anybody remembers that. And they went out of business, of course, because it wasn't sustainable. But one of the other things that I haven't really heard discussed much is the issue of archiving. And I know the open access journals, for the most part, are online, and they may or may not produce a print edition at all. And digital archiving is problematic, especially if the journal goes out of business. There's no paper copy of it anywhere, and the digital files might not be readable in 10 years.
Well, that's certainly a concern. Hopefully our journal will be in business 10, 20 years from now, but there's no guarantee of that. I think that's certainly a legitimate concern.
I guess you could argue that libraries could burn down too, right?
They could, but if you've got
so many copies all over the world,
let's say the Journal of Virology goes out of business, there are going to be hundreds of, or perhaps thousands of copies of that kicking around in basements, and it's paper, which people do argue that it decays over time, but it decays over a period of centuries rather than months.
Yeah, no, this is a big issue. I think it needs to be addressed.
Because we are, I mean, just so we're clear on what's at stake, this is the scientific heritage of humanity.
Right.
That is being stored in these journals.
This seems like might be something that NIH should actually take a look at, and if if the journal looks like it's going to go under, it wouldn't be a big deal to allocate some space to basically download all the articles and store them. It's just memory. And then
that's actually something that I thought was a really positive move with the establishment of PubMed Central.
Yeah.
Making that all in a common depository that is maintained by the government, which, say what you like, they probably will still be around in 20 or 30 years.
Well, if NIH isn't around, I don't think anything's going to be around. So, yeah.
Right.
And in fact, as I mentioned earlier, the papers that we publish from the NIH go into that, and we were required to deposit them there, and they'll be there hopefully forever.
Right. Yeah, I think the storage, that's a great idea to have someone take care of it, because storage is cheap now, and there's no reason why there shouldn't be a central repository. I mean, that would require the commercial journals to allow that to happen, but I'm not,
right.
And I don't know how that would go. Science, Nature, Cell, and so forth.
Actually, it'd be interesting to know what the statistics are on scientific journals that have actually gone out of business. I offhand don't know of any, but there might be some, and it'd be interesting to know what happened to their collections if that did happen.
Sure. Well, and I think we're going to see more, not just on the open access side, but I think we will see closed access journals in many cases going under, particularly if they're relying on very high subscription rates to sustain themselves. Yeah. I think that business model is going to get some stress.
Yeah, I mean, I think the move towards open access, I think, will keep the pressure on the conventional journals.
Right.
I don't know. The costs of publishing obviously vary depending on what you do. So my impression is that the PLOS journals have a lot of staff. They write all these summary type articles, review articles, and perhaps all that isn't needed. The bare bones is what you need. You need a staff to handle reviewing, and then you need the server to hold them. And you could argue that you don't even need to lay out pages. You just take the manuscripts as they're submitted and put those online. So why do they have to, be a certain format? And then that could be the cheapest possible way to run a journal. So do you need all the other stuff? That's the question.
Well, we have an editorial office that is a handful of people based in Basel, Switzerland, and they manage a lot of the email correspondence between reviewers and authors and myself and so on. And they also do the formatting of the articles when and the posting them online. But it's not a big effort. It's not a particularly expensive effort, and they run a handful of other journals as well.
Yeah, I think if you do a bare bones approach, you can. The work is still good work either way. It's just a matter of how you present it.
Well, I think actually the audience has already spoken on that issue. The reason you see, and here I should go ahead and disclose, this is how I pay a significant amount of my bills. If you look at the trend with Nature and Science and Cell, all of those journals, none of them have reduced their extra content or staff for that sort of thing in recent years. They've all increased it. So people are reading the news section and the news and views section and the analysis section of Nature and of Science. And if they weren't, the advertisers wouldn't be buying space in those areas. So and from talking to scientists, I know that that a lot of people do read a lot of these articles because it's a way of keeping up with things outside your field or in closely related fields. So I think there is a real value added there that's going to be missed if you have just this minimal pure text, poorly laid out, poorly edited and written. I mean, not to harsh too much on scientists' writing skills, but the reason I'm employed is that on average, a lot of researchers are not that great at communicating the work that they've done.
Yeah, nor do they want to take the time to write something that's generally accessible and make nice figures and all that, right?
Right. Nor do we want them doing that. I mean, if your talent is in the lab, then you should be in the lab working.
But scientists do write review articles, right? Which is probably appropriate because they know the field.
Sure. And news and views, which are a little more newsy. I mean, I don't think anybody has yet suggested that we completely eliminate the sort of old-fashioned conventional journals, Cell Science, Nature, et cetera. I think those serve very useful function. But I think this is something in addition to that I think provides people with alternatives.
So the main source of income for those journals, I presume, is advertising, right?
Well, the main source of income for a lot of journals is advertising. I don't know what the case is with viruses, but I know in PLOS's case, they certainly bring in a lot from advertising, and between that and page fees, those are the primary revenue streams. And at subscriber-driven journals, it's also the same. They make some from subscriptions, but the advertising and other income from that is going to be a major source.
So we don't actually have any advertising, so basically the publication fee will be sufficient to cover the cost of keeping the journal going. And again, of course, the editorial board, the reviewers, myself are all working free of charge, and so it's really just the editorial office that does the layout and so on. That's where the actual employees are.
But it seems to me that you could. Well, I don't know, actually. This is a question. Could you sustain an open access journal on advertisements alone? Or is that not possible?
I think you could. You can sustain. You can certainly sustain magazines on advertisements alone, and you can sustain even a research journal, Nature Methods, comes to mind, that is sustained as far as I know. It's driven by advertiser revenue. I don't know if Nature Methods charges a page charge, but I do know that they do not charge subscriptions.
So do you think, it sounds to me that both of you actually don't believe that all science journals should be open access.
I would love to believe that all science journals could be open access. I don't know if that's going to be feasible.
But I don't think Science Cell Nature will ever go that route, right?
It would be very difficult for them.
Why?
To make that transition.
I think they're serving, to some extent, the function of something like a Time magazine or Newsweek, where they have not only primary research publications, but also a lot of news and a lot of advertising. And I think people will continue to pay for that. And I think that's perfectly fine.
And the cost of publishing a Dead Tree edition of Nature is formidable. You certainly don't want that offloaded onto page charges, or nobody would be able to afford to publish in Nature. So they, just for that alone, I mean, to print and mail all the copies of that journal worldwide costs a fortune, and they need subscriptions to be able to drive that. So I think certainly until we get to a point where people are willing to universally do without paper copies of the journal, the subscription-driven model is going to be necessary for publications like that.
Well, I certainly don't need paper copies anymore. So.
Oh, I don't either, but I know, they can't just pull out of that.
Sure, I understand. But eventually they probably will. They will just go to an online model, right?
That's likely. Based on what's happened with the newspaper industry, I would say it's likely that we're going to see the end of paper magazines and even paper scientific journals.
The problem is that I understand these arguments for Cell and Science and Nature continuing as they are, but in the end, the papers are still not accessible to most people. And what do you do about that? I don't think that's appropriate. If you have a sick child, for example, and you're trying to read about diseases, you could go to a library, but they're not going to be around forever. So what's the solution to that?
I don't have one.
Well, I
mean,
guess
it's what I would argue is that the situation is a heck of a lot better now than it was even 10 years ago. So you're complaining about something that's not quite done yet, that the access to Nature or Science or Cell is limited, but it is still much greater than it used to be. And I think the trend is going to be toward more and more and more openness in the distribution, especially of important papers. And most of these journals have now already done this, where the really important hot stuff is usually accessible, or at least in some cases accessible. They'll make an exception and put it open access or something.
Mm-hmm.
I think also the trend will emerge, as it already has at the NIH, that the publicly funded research is made available, open access to everyone, regardless of where it actually gets published. So that's true now, again, of everybody here in the intramural program, and will probably become the case for extramural NIH-funded research.
Right. And to be clear, the folks I know at places like Nature and Science would love to be able to put this stuff out there for free. They just can't afford to. So the real issue comes down to, unfortunately, money. And say what you like about a big business, they do have to stay in business to continue publishing these journals. So they have a very real concern that the open access model may not be sustainable. And I think until that's proven, they're certainly within their rights to keep the access somewhat closed.
Well, as long as scientists keep submitting papers, they'll survive.
Sure.
And as you know, when you come up for tenure, it depends on how many Cell, Science, Nature, etc. papers you have. And that's one of the big issues, I think, is that these open access PLOS has now become quite respectable. Discussions I've heard, it's still some question whether it will be around still in 10 years. So, when tenure decisions are made, I'm not sure how much PLOS plays into that.
Yeah. Well, if you look at the impact factors of the PLOS journals, they're actually really quite high, actually, particularly journals like PLOS Biology.
Mm-hmm.
PLOS Pathogens is very strong, too. So I think these journals are really emerging as real competitors for the Cell, Science, and Nature, or at least viable alternatives.
Right, if they're still around.
Yes, that's the thing. I mean, we don't know the answer. I hope they are, because I think it's terrific that everyone can access the papers. And, I mean, we always complain that people don't understand science. And part of it is letting them read it, and they can't at the moment.
Absolutely.
They can't read a lot of it. So, kids in school can't get papers just to read. And I know they might not understand it, but they could make a start and start asking questions. And so it seems to me to wall off all that research is just not right. But then again, I think everyone should have access to health care without restrictions too. So I'm just the most unrealistic person that ever walked the face of the earth. But I myself have wonderful access to all the journals I would want here at Columbia. But as soon as I go home, I can't get most of the papers. And I would like to work from home because I have to go to my PC and I can do that.
That's right.
Maybe I could access. Actually, I should try that. I could probably.
I bet you could do that. I bet you could do a little wormhole in.
Yeah.
With my PC.
Because if I can log on to my computer, I should be able to go through its network to the library and get all the papers that I want.
Yeah.
Well, I hope Viruses does well, Eric.
Well, thank you.
Yeah. I mean, are you happy so far? Do you feel that it's going in the right direction?
Well, yes, I think it is. And again, I think really, going back to the topic of whether a journal will survive, I think really the key is to start with a strong editorial board and solicit authors who are first rate and subject whatever is submitted for publication to a very rigorous peer review process, publish high quality work, and I think that's 99% of the battle right there.
I think that's the key. If you publish high quality work, you'll be fine.
That's right. If it's junk, it doesn't deserve to survive.
Yeah, you quickly get a reputation. They just publish nonsense. So,
yeah. Although
the best journals sometimes publish garbage, as you know, but they do.
Some of the best do. Yeah.
They do publish a lot of good stuff as well. Well, I'll send a paper in. Not that's not going to help you very much, but I'd be interested to publish there because I like.
I would appreciate that.
The more options there are for publications, the better off in the long run.
I agree.
Well, I would love to talk about HIV, but I think we need to move on to email, and that way keep it below an hour and a half, because I don't know if you noticed, Alan, our TWiVs are getting longer and longer.
Yes, I did notice that.
And I don't want to go over, but if that's okay, I'd like to have you back another time, Eric, and we can talk about HIV and antiviral drugs.
I'd be happy to do that.
Because I like the sound of the old classic phone line. Alan, it reminds me of the old interviews, doesn't it?
Yeah, it's like on NPR when they call somebody up.
Yeah. So you sound good on the analog phone there. So
thank you.
So let's do some email. We have a couple here. Andre wrote, "Hi, Vincent, Dick, and Alan. I've just finished my PhD thesis on the fusion protein of respiratory syncytial virus. As a virologist, I particularly enjoy your show. The virological community is widely scattered across the globe and gets to meet only on very few occasions at international conferences. But listening to your podcast makes me feel much more connected to the rest of the scientific world outside my city, Hanover, Germany. This, I think, is really cool, and it's a great contribution made by you. Anyway, I wanted to point out a website from the American Society for Cell Biology, which offers a series of advanced seminar lectures for download. David Baltimore, for instance, talks about HIV. It's ibioseminars.org on iTunes U.
Cool.
I always like to hear from virologists. It's nice that they are listening to us, which means that we're putting TWiV at just the right level to get not only people who don't know much about viruses but would like to, but virologists as well. So that's great. Orion wrote, "Hello, I would like to say that your podcast has made me very interested in virology. I had chosen biochemistry as my major after taking an intro course in molecular biology. I was originally a chemistry major, and after taking molecular, I found that the cellular processes involving DNA, RNA, and proteins was the most fascinating thing I had ever learned of, and therefore decided to change. Up to this point, however, I had no idea what I was going to do or research. I've been listening to your podcast for a couple of months now and found what you have to say about viruses very compelling. I seriously think I may go into virology."
Excellent.
Great.
Hope he does.
I have a couple of questions. I have hereditary pancreatitis, which is caused by a mutation in the gene for trypsinogen. I have read that viruses can be used to introduce DNA into genomes using retroviruses and wondered if this was the type of thing you might do in virology. Finding a treatment for my own disease was something I had considered focusing on as a future scientist. Also, I started listening from the beginning of your series of shows a couple of months ago and just now heard of your contest and book giveaway. I was wondering if the contest was still on or if there are still books available. Thanks for the great podcast and helping by introducing me to a very exciting field. So, Eric, what about using retroviruses to cure genetic diseases? You know anything about that?
Well, there's certainly a tremendous amount of research being done in that area. There are some pitfalls in terms of disease induction caused by the integration of the retroviral DNA into inappropriate sites in the human genome. But I think so there are other viruses that don't integrate that are also being pursued as gene therapy vectors. But I think certainly retroviruses have a lot of potential for human gene therapy.
Yeah, so you can do that in virology, and you could train and go work on projects that involve doing that. Absolutely. As far as the book giveaway goes, that's done, that contest, but we'll do another one in the future, so keep listening and you'll find out when it comes up again. We're talking about giving away copies of my textbook, Principles of Virology. We had a link contest a few months ago, but stay tuned and we'll do some more. And then we have a letter from Jim, which I can't read in its entirety, but I want to start, and we'll read it over the next few episodes. Vincent, Dick, and of course Alan. I finally listened to all the TWiV podcasts up to number 31. Excellent and intriguing presentations. I apologize for the following message and variety of topics, but it just kept growing as I listened to all those podcasts, so it took a couple of weeks to put everything into a reasonable format. I'm a layman, 68 years old, retired nuclear power plant operator. After an Army career as a criminal investigator specializing in technology, completed a one-year fellowship at Armed Forces Institute of Pathology in 1975. Met my wife there. Incidentally, the AFIP was built to withstand a nuclear attack before the H-bomb made such protection unlikely, so the entrance is flanked by two large bank vault doors.
Cool.
I didn't know that. Your discussions are intellectually stimulating, and much of it impacts my wife and I, our family, neighbors, and friends, as aging individuals and stewards of our environment. Some impacts are closer than others. We have a septic system and are acquainted with Dick's Schmutzdecker. You remember that, Alan?
Yes, of course.
And once read that the gravel filter underlying the drain pipes somehow neutralized viruses in the effluent. Does that make sense? Are there precautions one should take when pumping a septic tank?
Breathe through your mouth.
Septic tanks, Jim, are full of enteric bacteria and viruses. They've been shown to transmit all of those, and in fact, there are regulations for installing septic tanks, which are very strict so that they don't contaminate the water supply. And it's been shown that viruses can be transmitted from effluent.
Yes.
And it turns out that there are a lot of studies that have been done. The gravel filter is actually sand under the tank that filters the bacteria and viruses. There has to be a certain thickness of sand underneath the tank and underneath the pipes. And the viruses actually stick to the sand, the silica. And many studies have shown that after so many inches of sand, the viruses are removed. So they're not actually neutralized, they're just filtered out. And if the sand isn't the right thickness, you don't get effective filtering. But you can find some very interesting articles, and I'll post a couple of links for you so you can look into that. Birds at our feeders are sometimes obviously afflicted, and we've been watchful for possible West Nile candidates since it was identified as a hazard. We've wetlands and small pools of stagnant water when rain is plentiful and resulting varieties of mosquitoes. Yeah, you have to watch for those standing water pools. And as Dick says, the birds are dying, and depending on where you are, if you find a dead bird, you could always bring it into your local health agency, and they might check it out for West Nile.
Yeah. Well, actually, you're probably better off if you find a dead bird just reporting it to your local health agency.
Oh, I think you should walk in the door, don't you, Alan?
You will get a lot of attention that way.
Yeah, you probably shouldn't touch it, right?
Probably best not to pick it up.
This summer, 4 skunks, 2 of which were pregnant, were trapped from under a neighbor's outbuilding. A bachelor living either side of us had started putting food out for them over a year back. You guys aren't bachelors, are you? No,
No.
I'm not. Rabies have been well established around us for 10 years. Well, skunks can get rabies, but so can most mammals, and they get sick from it. So you would know if the skunks were rabid. They behave very strangely. We've picked 100 ticks off our dog this season despite a flea collar and topical anti-tick medications. My wife was treated for spotted tick fever and ehrlichiosis several years ago, making us even more sensitive to protecting ourselves. Yeah, ticks are really excellent places for microbes. And I just went to a talk recently where a postdoc was collecting ticks from Connecticut and Long Island and doing PCR to look for viruses and bacteria. And, of course, they're full of Lyme bacterium, and there are some viruses as well. So be careful for them. And we have more types of biting insects than any place I've been, from chiggers to horseflies, black widows, brown recluse spiders. We live on the James River, with a varying salinity depending on rainfall and tidal action, in which at least one serious affliction can be contracted by handling marine life harvested as food. I can't recall if it's viral or bacterial. Your team approach on the podcasts are great. You added a younger generation with Mark Pelletier and his perspective. Someone in the 18 to 25-year range might extend this bridge to younger listeners, a bright high school senior or undergrad student. Mark's son was so good at computer work, perhaps even someone that young can contribute by listening to a podcast and asking questions. Yeah, I think that's a good idea. I don't know. During the school year, it's hard because we do this at 2 p.m., but maybe over the summer I can get an interested science student in to sit by and ask questions. That would be cool, wouldn't it?
Yeah, that's a good idea.
Then he says, and I'll wrap it up with one more sentence, each podcast should be used for extra credit or teaching tool in high school onwards. Wish I could suggest a contest involving computers or cell phones or Twitter that would advance virology. Yeah, well, we work on it. We're spreading virally. But extra credit is a great idea. I would like to figure out how to get TWiV into high schools or some kind of science curriculum. It's kind of long, I guess.
Starbucks.
We're in Starbucks, that's right. Well, next spring I'm teaching an undergraduate virology course here at Columbia, and I'm going to include in some way TWiV. I haven't figured it out yet, but I will. So I'll stop there. He's got a lot more we'll read on future episodes. And I thank you for that nice email, for listening to all our podcasts. And in fact, Jim sent me an audio file the other day, an MP3 letter, which we'll read next time.
Excellent.
It'll be good. I appreciate that a lot. And that brings us to our science picks of the week. So, Alan, what you got?
Yeah, I've got well, it's a website. It's actually a person, Forrest Mims. We'll have a link to his website in the show notes. And I guess I came across Forrest a number of years ago. He's best known as the author of a book called Getting Started in Electronics, which is about what it says on the cover. It's probably the best introductory electronics textbook ever, and I think it's the best selling. But he actually is a really interesting guy. He's done a lot of amateur science that has risen to the level of professional science. So as one example, I think about 20 years ago, he was puttering around with some LEDs and circuitry and playing with electronics, and he developed a sensor that would sense the amount of sunlight coming down on the sensor. And he then proceeded to use this to take daily observations of the sunlight every day at noon, which gives an indication of the level of atmospheric pollution, of aerosols and haze. And he's been doing this for a couple of decades now, and it turned out some of the results that he got didn't jive with the data that NASA was getting from their Earth science satellites. So initially he contacted them and, was trying to figure out how to calibrate his device, and what transpired was that in fact the satellite was wrong. So NASA has now used his data to calibrate their satellites. And this has gone on. He's built this into a whole career as an atmospheric scientist, who is essentially self-taught. And it's kind of an example of the kind of thing that one can do if one is motivated enough to pursue the science. It's in many cases right outside your door. And while I don't advocate culturing viruses in your basement, depending on the viruses, that might be a good thing to do. So it's a really neat story and an interesting person who's done some absolutely fascinating science, just by following his whims.
It's very cool. I see, right? I'm on his website. What am I doing? Building a day-night cloud detector using a thermoelectric module. He's got great photos here. So this is kitchen science, right, Alan?
Yeah, it's kitchen and backyard science.
That's great. That's very nice. I like that. Yeah, it's hard to do with viruses. Or you could do it with phages, which aren't really dangerous. You could do plaque assays and growth curves and so forth. You could filter them through the sediment in your septic tank and see how they're inactivated and so forth. Well, that's good. Thanks. Forrest Mims, Getting Started in Electronics is his book. Excellent. Thank you very much. My pick is a little book called The Machinery of Life by David S. Goodsell. And David Goodsell is a scientist and artist out at Scripps. And you might know him online from his wonderful illustrations of molecules. If you go over to the Protein Data Bank, there's always a molecule of the month, I think, and he draws the pictures. He's a very good artist, and this book is all about molecules. Gives you a sense of how big things are and how they relate to one another, and he draws them all out. And molecular illustrations, a little paperback book worth looking at. And a lot of listeners have said they don't understand how big this is compared to that, how big is a virus compared to a cell or a bacterium. This book will give you an idea of that, The Machinery of Life. So, Eric, what have you got?
Oh, I didn't know I was supposed to pick something.
Is there anything you liked? Did you read a book when you were young that made you want to go in science?
Oh, I had a lot of science books. I was actually very interested in meteorology and very interested in geology. So I have a lot of books that I actually gave to my children a few years back that show all kinds of interesting science-related things, and I think they also really enjoyed them.
Well, anything science is good, but we'll let you off the hook. Next time we'll let you have a pick.
Next time I'll be better prepared for that.
No problem. We're a member of sciencepodcasters.org and promednetwork.com. Those are two websites where you can find other good science podcasts. Eric, thanks for joining us. Really appreciate it.
Yeah, thanks for having me.
I am going to put a link to your website at NCI, which I found online. Is that okay with you?
That's certainly fine.
It's home.ncifcrf.gov, et cetera, et cetera.
Yeah.
Alan, thank you again for joining us.
Always a pleasure.
Now, let me get this right. Alan Dove on Twitter and dovedocs.com.
That's right.
Is your blog.
That's right.
I got it right after several weeks. And you can follow TWiV on Twitter, PROFVRR. TWiV has a website where you can find all the show notes for all the shows and all the links, twiv.tv. And you should also send us your questions and comments to twiv@twiv.tv. And if you'd like, you can send an audio comment as an MP3 file. As I said, we got our first one, and we'll play it next week. We'll be back next week, of course. In the meantime, please subscribe in iTunes if you haven't done so already. If you don't use iTunes, you can always listen to us at twiv.tv. You've been listening to This Week in Virology, podcast all about viruses. Thanks for joining us. Another TWiV. It's viral.